Is it audible? It is. Okay. So we are in very interlinked part of the biological classification with regard to what is fungi. Earlier we discussed cellular, what is the filamentous, and what basis it classified. That is the name, morphology of the mycelium, that is aseptate or septate, and it is fruiting body. It is classified into varieties, the Phyco, Asco, and Basidiomycetes. And nature of the mycelium, whether it's a branched or it is unbranched, septate or aseptate, like this. In this Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. There is a gradual reduction of sexuality from Phycomycetes to varieties Deuteromycetes. In this Rhizopus, Mucor, Albugo, and Pythium. These two are additionally important. Albugo, that causes varieties white rust on varieties the mustard, and in case of Pythium, that is the fungal cell wall having variety cellulose. So fungal cell wall usually it is the chitin. In some cases it is also cellulose in case of Pythium. Okay, Rhizopus, Mucor, Albugo, Pythium in case of Phycomycetes. In case of Ascomycetes, Penicillium, the Phaeacia, Aspergillus, Morels, Truffles, Neurospora. So Penicillium antibiotic, Claviceps, ergot of rye, ergotamine, alkaloid obtained. So Claviceps causes what it is ergot in case of the rye plant. And Aspergillus, morels and truffles, which are delicacies, add flavor to varieties, the chocolates, the root fungus it is morels and truffles, the costliest mushrooms it is morels and truffles. And the plant genetics, biochemical and genetical studies, it is used. Cinderella of the genetics, it is nothing but varieties, Neurospora. So Penicillium, Phaeacia, Claviceps, Aspergillus, Morels, Truffles, Neurospora comes under varieties, that is Ascomycetes. And in case of Basidiomycetes, Puccinia, Stylago, agaricus, Puccinia, Stylago, agaricus, Lycopterodon, that is the puffball. So Puccinia, Stylago, agaricus, Lycopterodon. In case of it is Basidiomycetes. Polyporous. The question is going to be like this. So number of the fungi species the given as example. You have to find out which is Ascomycetes, which is Basidiomycetes, which is deuteromycetes. And in case of deuteromycetes, Alternaria, Coritotrichum falcatum. And Trichoderma. So in case of Phyco, Asco, Bacillio and Deuteromycetes. And earlier we said in case of Phycomycetes, plasmogamy followed by karyogamy, what is delayed? It is meiosis, it is delayed. If you see in case of Phycomycetes, gametangia will fuse. Suppose it is having 40 plus nuclei, 40 minus nuclei. Plasmogamy followed by it is karyogamy. The karyogamy results in a structure, it is zygospore. So 40 haploid, 40 haploid, 40 it is diploid nuclei. The 40 diploid nuclei undergoes meiosis, form 160 nuclei. Of this 160 nuclei, 80 plus, 80 minus, that is going to form the spores. What do you call? Meiospore. So, which event is delayed? It is meiosis. It got delayed in case of Phycomycetes. Plasmogamy immediately followed by karyogamy. In case of Phycomycetes, the delayed it is meiosis. In this multi-nation, what do you call it? Is xenocytic condition in case of Phycomycetes. And in case of Ascomycetes, it is parasitic, saprophytic and dung-living fungi. What do you call it? Is coprophilous. Mycelium, it is a branched, septate and ascogenous hypha formed. This ascogenous hypha, it is dikaryotic binucleate haploid. So here plasmogamy, that is somatogamy. And what is delayed? Karyogamy delayed. Then karyogamy occurs immediately followed by meiosis. If karyogamy occurs immediately followed by it is meiosis. But what is delayed? Karyogamy delayed. What results in dikaryon? That is n plus n condition. But not it is 2n. N plus 1, that is dikaryotic haploid mycelium. And here fruiting body, what do you call? Ascocarp. Fruiting body, it is ascocarp. The ascocarp, it is spherical without any opening, it is cleistothecium. And flask shaped perithecium, saucer shaped, it is apothecium. So, non-porous fruiting body is cleistothecium. And flask shaped with apical opening, it is perithecium. Saucer shaped, it is apothecium. These fruiting bodies having what it is? Ascus. Sac-like, it is ascus. Inside the ascus, how many nuclei enter? The 2 nuclei will enter. So, karyogamy occurs in ascus. Meiosis occurs in ascus. Results in ascospore. Origin, it is endogenous. So, karyogamy, meiosis occur in ascomycetes in ascus. So, inside the ascus, what are formed? Ascospores. Suppose it is fruiting body, cleistothecium, it is ascocarp, fruiting body. It is having ascus. So inside the ascus, karyogamy and meiosis occur, results in vertex. Ascospores which are haploid. Sexual haploid endogenous spores which are ascospore. Ascospores are nothing but meiospore, resulted because of what it is, meiosis. So in this, conidiospores which are asexual, which are exogenous. Exogenous asexual spore, endogenous sexual spores are present in ascomycetes, conidiophores. These are conidiospores. It is conidiophores. The conidiophores are multicellular, branched. At the apex of this conidiophore, a bottle-shaped structure, it is called sterigmata or phyalides. On phyalides and sterigmata, what are present? Conidiospores or conidia, which are unicellular. Conidiospores or conidia. Sterigmata, conidiophore. It is fruiting body, Clistothecium, ascocarp, ascus, and it is ascospores. So, karyogamy immediately followed by meiosis in karyogamy and meiosis simultaneous. Karyogamy and meiosis occur one after other immediately in A. Phycomycetes, B. Basidiomycetes, C. Ascomycetes, D. The question number 1. Question number 1. Karyogamy and meiosis occur one after other immediately in it is asco and what it is basidiomycetes. Meiosis delayed in case of what it is phycomycetes. And basidiomycetes, what do you call it is? Club fungi. Why club fungi? Because club shaped basidium. In this first two primary mycelia, the two primary mycelia, suppose it is +4, it is -4. The two primary mycelia undergoes somatogamy. The two primary mycelia somatogamy. Which cell is formed? Dikaryotic cell. Develop into which mycelium? Dikaryotic mycelium. Developed into fruiting body. What do you call it is? Carp. In basilocarp, what is formed? Basidium. In basidium, the two nuclei fuse. What? Karyogamy. And karyogamy followed by meiosis results in basidiospore. Here, how many kinds of basidiospore? Two kinds of basidiospore. Sequence A, B, C, D, E, F, G, H, I, J. Two kinds of basidiospore plus and minus forms two primary mycelia. Two primary mycelia undergo somatogamy, results in dikaryotic cell. What is delayed? Karyogamy is delayed. Which mycelium is formed? Dikaryotic mycelium is formed. What is the fruiting body formed? Basidiocarp. In basidiocarp, what is formed? Basidium. In basidium, two nuclei migrate, plus and minus, results in karyogamy, that is diploidization, followed by meiosis, results in how many kinds of basidiospore? Two kinds of basidiospore. How many basidiospores which are formed? Four basidiospore. 2 plus and it is 2 minus. Okay. This is the sequence. Suppose in basidiocarp, there are 600 basidia present. 600 basidia present. A total nuclei involved in karyogamy. Number of synkaryons formed. Number of synkaryons formed. Number of reduction divisions. Number of basidiospores. A number of plus spores. Question number 3. Question number 3. Answer. In basidiocarp, there are 600 basidia present. If 600 basidia present, total nuclei involved in karyogamy. A, B, C, D, E. How many? 300 nuclei. How? In each basidium, how many nuclei which are involved? 2 nuclei which are involved in each basidium, plus and it is plus nucleus, it is minus nucleus. 600 bacilli are present. So in each basidium 2 nuclei, how many nuclei which are involved? 1200 nuclei which are involved in karyogamy. Synkaryons formed in this one synkaryon. So how many synkaryons are formed? 600. How many reduction division in each one? One reduction division. How many? 600 reduction divisions. Number of basidiospores. Each basidium having 1, 2, 4. 2400. Number of plus spores 1200. Okay, got cleared. Now got cleared? All. In one basidium, 2 nuclei migrate plus and minus. So each basidium, how many nuclei which are involved in karyogamy? 2. As the 2 nuclei will fuse, one synkaryon is formed. Okay, in 600 basidia, 600 synkaryon. One synkaryon, that is the diploid nucleus, one meiotic, 4 nuclei which are formed. As 600 into 4, 2400. So 2400 spores are formed. In this 1200 plus spores, 1200 varieties minus spores. How many kinds of the spores are found? 2 kinds of the spores, plus as well as varieties minus. Okay. In case of varieties, the basidiomycetes, there is the sequence. And in case of asexual spores are generally not found in basidiomycetes. So sexual spores are exogenous. Asexual spores are generally not found in basidiomycetes. Asexual spores are exogenous. Sexual spores are endogenous in ascomycetes. Asexual spores are endogenous in phycomycetes. Sexual reproduction not at all discovered in case of deuteromycetes. That's why it is fungi imperfecti. That means sexual stage was not discovered or it was absent. That's why it is fungi imperfecti. So there is a gradual degeneration of the sexual mechanism from Phyco, Asco, Basidio, and Deuteromycetes. There is a gametangel fusion in Phycomycetes. Full walls are uniting in Asco and Basidiomycetes. And sexual reproduction itself is absent, not discovered in case of Deuteromycetes. In this, vegetative mycelium asexual stage given one name placed in Deuteromycetes. If sexual stage discovered placed in classes of varieties Deuteromycetes first. If linkages established moved out of Deuteromycetes, if any perfect stage discovered moved into Asco Basidiomycetes, not in case of Deuteromycetes. So vegetative asexual stage given one name, sexual stage varieties other name. If sexual stage discovered placed in right classes, and if linkages are any established moved out of Deuteromycetes, and if perfect stage were discovered moved into Asco and Basidiomycetes. In this example, Alternaria and Coritotrichum as well as Trichoderma in case of Deuteromycetes, which are mineral, helps in case of mineral recycling. That is, the letter into humus, humus into what it is, the minerals. So, branched septate mycelium in case of Deuteromycetes. Phycomycetes, branched septate multinocytic Phycomycetes. Ascomycetes, binucleate. Basidiomycetes, binucleate, which is septate. Deuteromycetes, uninucleate, and it is septate. So, sexual reproduction not discovered and it is absent in case of Deuteromycetes. So, in case of biological classification, lichens, virion, virioid, not placed in case of Whittaker system. And before that, Plantae and Animalia. In case of the Plantae, it is autotrophic, but it is the parasitic forms which are present and insectivorous plants are present. Parasitic form, partial root parasite, it is Striga. Complete root parasite, Rafflesia, Orobanchi. And partial stem parasite, Loranthus. Complete stem parasite, Cuscuta. So, which are partial or complete parasite, but plants which are autotrophic and holophytic mode of varieties nutrition. And in case of Animalia, holozoic mode of varieties nutrition. And in higher forms of varieties animal sensory and neuromotor system, it is the present in case of varieties Animalia. And food reserves are present in the form of varieties the glycogen in case of Animalia. And the lichens, virion, virioid, and viroid and prion did not find its place in case of Whittaker system. And lichens, which are amalgamation of varieties, algae plus fungi. And this association may be parasitism, maybe it is symbiosis. So, algae provide varieties, it prepares the food material. Fungi provide varieties, water, mineral, and shelter. Algae prepare varieties, food material. So the relation in case of the lichens may be helotism consortium. Some scientists viewed it as a helotism master-servant relation. Some scientists viewed as what it is husband-wife relation consortium. So anyhow, the amalgamation of the algae and fungi results in what it is the lichen. And lichens which are pioneers of what it is hierarchical succession, it did not find in a places. That means it is indicators of what it is the pollution. That means it did not find in a place where it got what it is the polluted. In a clean environment it is present. That's why it corrode on the rock surfaces, secretes carbonic acid, disintegrate rock, makes the way for what it is mosses. That's why lichens are considered as pioneers of hierarchical succession. Phycobiont and mycobiont component present in case of what it is lichen. So, and these viruses, with regard to scientist Louis Pasteur, Ivanovsky, Beijering, Stanley. Viruses which are a bunch of genes always in a search for what it is the host cell, where it attack what it is the host cell, multiply and leads to lysis of that particular host cell is the duty of what it is the virus. So, viruses are obligate intracellular parasites. Viruses are more than lifeless inert molecule. So, viruses more than lifeless inert molecule. The statement right or wrong? The fourth question. More than lifeless inert molecules. Question number 4. True or false? Yes, more than lifeless inert molecule, because it is having the nucleic acid, it can able to mutate. It utilizes metabolic machinery of the host. That's why. Okay. And Louis Pasteur consider it as what it is the venom or it is the poison. Venom or poison. Ivanovsky observing what it is the tobacco mosaic disease, the viruses which are filterable through bacterial filters according to Ivanovsky. And contagium, it is viruses which are filterable, tobacco mosaic virus. And Beijering, contagium vivum fluidum. And the milestone contribution by Stanley, viruses which are crystallized first time, the crystals who got it is protein. Tobacco mosaic virus crystallized by Stanley. What he got? Proteins. Beijering, contagium vivum fluidum. Ivanovsky, viruses which are filterable. Louis Pasteur, virus is a venom or otherwise what we call it is poison. So more than lifeless inert molecules. And nucleic acid plus protein. What do you call it is virion. So plant viruses having RNA, animal viruses having DNA. In general, plant viruses having RNA, animal virus it is DNA. RNA usually single-stranded, DNA usually it is double-stranded. So usually it is double-stranded because the AIDS virus it is the single-stranded RNA. It is also present in case of the human virus. So in general, DNA it is double-strand in animal viruses. In general, in plant RNA it is single-stranded. So complete virus particle, what do you call it is virion, or otherwise what do you call it is virus. And the protein coat, what do you call it is capsid. And subunits, what do you call capsomeres. And in case of animal virus having DNA, or some cases for it is RNA, that is nucleic acid plus capsid plus envelope, that is having carbohydrates, lipids, proteins. So animal viruses DNA or it is RNA having capsid surrounded by it is envelope. Envelope having carbohydrate, lipids and protein. So a novel corona virus, the general use, the method as a preventive method, it is the washing our hands with detergent. The detergent is going to act on what it is the lipids. Lipids will be what it is losing its what it is its character. So detergent what it is the lysis of the membrane used in case of the biotechnology, as it is no membrane. So lipids or it is destructed or denatured by using what it is the carbohydrate, lipid, protein present in case of what it is envelope. So as lipids are going to be disturbed, so the virus cannot what it is survive because it provide what it is stability to what it is the total the virus. So envelope present in extra in case of animal virus, that is RNA or DNA surrounded by it is envelope. So in case of animal virus, and prion abnormally folded protein. Neurological disorders, because of what it is abnormally folded what it is the protein. So mad cow disease and Creutzfeldt Jakob disease in humans. So transmitted through what it is the prion. So mad cow disease, the same it is the in case of the humans is CJK disease, that is Creutzfeldt Jakob disease. So virus minus nucleic acid, nothing but what it is the prion. Prions are infectious or it is genetic. Prions are infectious and genetic. Question number 5. Prions are infectious and genetic. Yes, it is false. Prions are only it is infectious, but it is not a genetic. Okay. The first discovered virus belongs to a plant that is under dash family. Question number 6. The first discovered virus belongs to a plant virus that is under dash family. Question number 6. Family of plant. Family of the plant. Which family? Tobacco mosaic virus. Tobacco belongs to Solanaceae. Solanaceae, it is a dicot member. Okay. And viroid. It is with less molecular weight than what it is the viruses, having only nucleic acid. Viroid infect only plants. Potato, spindle, tuber virus, citrus, exocartosis virus. Okay. Potato, spindle, tuber virus, citrus, exocartosis virus, which are viroid, which infect only it is the plants. As viroid infect only the plant, viruses infect what it is. Okay, all viroid infect only the plants. Okay. Shape of TMV, symmetry of TMV, shape of T2 T4 phase, symmetry of T2 T4 phase. 8, 9, 10, 11. Shape of TMV, symmetry of TMV, shape of T2 T4, symmetry of T2 T4. 8, shape of rod shape, symmetry helical, shape of T2 phase is tadpole, symmetry polyhedral. The bacteriophages which are polyhedral symmetry. Okay. Tail fibers also called dash. 13. Dash is ratio of tail fibers to tail pins to end plates. Question number 12. Yes, what do you call prongs? Dash is the ratio of tail fibers to tail pins to tail plates in case of T2 phase. 6 tail fibers, 6 tail pins, 1 end plate. Okay, 6:6:1. No, 6:6:1. 6 tail fiber, 6 tail pins, and what it is 1 end plate. Okay. Okay.